Good evening, everyone. Apologies for the slight delay. I was waiting for a video to show, but it's not going to, I think. Welcome to the second event in our Food for Thought series. I would like to acknowledge and pay respect to the traditional owners of the land on which we are this evening, the Ngunnawal people. It's upon their ancestral lands that the Australian Academy of Science is built. And as we share our own knowledge, teaching, learning, and research practices, may we also pay respect to the knowledge embedded forever within the Aboriginal custodianship of country. My name is TJ Higgins. I'm a fellow of this academy and one of the co-conveners for this series. Also joining me as the other convener is Dr. Emma Beckett, joining us tonight from the University of Newcastle on the central coast of New South Wales. Welcome.
Thank you so much, TJ. I am delighted to be convening this series with you, and I'm joining you today remotely from Darkinjung country. I too would like to pay my respects to elders past, present, and emerging, and acknowledge that sovereignty of this land was never ceded. Thank you all for joining us at our second event in the series that will take you on a tasty journey of nutrition, health, and food innovations. Tonight, we're going to be talking about gut health. I invite you to join the conversation on Twitter using the hashtag FoodThoughts21. FoodThoughts21. That's hard to say with a lisp. I'm going to have to think about that harder next time. For those of you who are watching online, you can submit your questions by scanning the QR code that will appear on your screen, or you can click the link on the page where you're watching this stream. For those of you who are in the Shine Dome, you can ask questions at the microphone during question time. Back to you, TJ, to get us started with gut health.
Thanks, Emma. I would now like to introduce tonight's first speaker, Dr. Claire O'Brien. Claire is an assistant professor and head of the host microbe interactions laboratory at the University of Canberra. Her research group focuses on understanding how disease impacts the gut microbiome and strategies to restore microbial ecosystem equilibrium, as well as the crosstalk between host and bacterial cells in health and disease. Please join me in welcoming Claire.
Okay, so good evening, everyone. Thanks for coming along tonight, and hello to everyone online. Thank you to the Academy for inviting me to talk about a topic that I love to talk about, the gut microbiome. So the gut microbiota comprises all of the microbes that are possible, so the bacteria, the archaea in the light blue, which look like bacteria but are actually evolutionarily distinct, the fungi, so the budding yeast cells that you can see in the middle there, as well as viruses that can infect us, as well as viruses like the robotic-looking virus up there, the bacteriophage that infect bacteria. Next. So you've probably heard that we're mostly microbe. So a few years ago, you may have heard that microbial cells outnumber our own cells by a factor of 10, but this has been re-estimated, and we now think that we're about 43% human. So we have about 30 trillion cells in our bodies, but that's opposed to the 39 trillion microbial cells that reside on or within our gastrointestinal tract. And it's even more striking when you look at this at a genetic level. So we have about 20,000 human genes, each of us, in our DNA, but there's about 2 to 12 million microbial genes in the microbes that reside on us. So pretty fascinating stuff. We are more microbe. And your gut microbiome comprises about 1,000 species of bacteria. And it's unique. So everybody's microbiome is unique. It's like a little fingerprint. Next. So this slide here just shows the distribution of the major bacterial phyla that comprise the gut microbiota. So they're the Firmicutes, the Bacteroidetes, and the Proteobacteria. And you can see on the slide that the gastrointestinal tract mostly comprises about 90% of the Firmicutes and the Bacteroidetes. And you can see that the microbiomes in other areas, such as on the head, are very different to that of the gut. So we see the green bacteria on the head, the cyanobacteria, and they actually use sunlight in a process called photosynthesis, but we don't have any sunlight in the gut, and that's why we don't have any in the gut. So our microbes are very well adapted to the gut environment, and they've evolved with us over time. Next. So there are many benefits of the microbiome. They train our immune system, they synthesise vitamins for us as well as amino acids and other things. They regulate cell turnover in the gut, and they even determine our response to most medications. So pretty fascinating. They also break down indigestible components of our diet, for example, fibre, and I'll talk more about this later. Next. So it's thought that we largely acquire our microbiome when we're born. There is some evidence to suggest that we acquire a small amount of bacteria in utero, but that's a bit controversial still. But the mode of birth matters as well. So those babies born vaginally will acquire the microbes in the mother's vaginal tract and on the skin in the perianal and vaginal region when they're born, whereas babies born by C-section will acquire microbes in the hospital environment, which are more pathogenic. So these microbes are also, the C-section microbiome is also associated with an increased incidence of asthma, eczema, and allergies, and so a lot of research is directed at the moment towards reseeding the microbiome, so it's more like the vaginally born babies. So by the age of 3, not only do we have a lot of personality like this little 3-year-old did, we've also acquired a microbiome that's very diverse. So by the age of 3, our microbiome resembles that of an adult microbiome in terms of its diversity, although there is a little bit more development that needs to take place. Next. So a number of factors will influence your microbial composition. Obviously, antibiotics will, because they're actually designed to kill bacteria, so they'll wipe out certain sections of your microbiome. But where you live also influences your microbiome. So unlike SARS-CoV-2, which is very easily transmitted, your gut bacteria is not as easily transmitted. So we know in studies that we've done in our lab that we have strains of a bacterial species called E. coli, for example, that we see in the human guts of Canberrans, but not those living in Paris. We also know that your diet will influence the gut microbiome, and I'll talk a lot more about that later. Chance encounters like a handshake will also influence what microbes establish themselves in your gut. So you might shake someone's hand, they'll have a bacteria on there, put your hand in your mouth, and it may or may not establish in your gut, but it may. And then, although we have a unique microbiome, we do know that we're more likely to share microbes with other household members, even the furry friends. Next. So, we think that a healthy microbiome is a rich and diverse one. And some of the measurements that we use to evaluate the microbiome include alpha diversity measurements. And these measurements take into account the number of species in our gut microbiome, but also the evenness of those species. So you can see on the left of the slide, there's a microbiome with the X, which is represented by 3 different species, but the pink bacterium is dominating in that microbiome. The next one along, we again have 3 species of bacteria, but it's more even, so there's 2 of each bacterium there. The next 2 microbiomes that you can see there both contain 7 species of bacteria, but the one on the left has an expansion of the pink bacteria, whereas the one on the right, with the tick next to it, is more evenly distributed. So we think that This is what a healthy microbiome looks like. It's diverse and it's quite even. You don't see an expansion of one group of bacteria over another. And the reason why we want a diverse microbiome is because of this idea of functional diversity. So if we had two microbes, take the top two bacteria in the nice diverse microbiomes, and they carry out the same function for us, say fibre degradation, if we lose one of those bacteria, then we're okay. We've got a sub on the bench that can take on that function. So we wouldn't have that in the less diverse microbiome on the left. Next. So another measurement that we commonly use to study the microbiome is beta diversity measurements. And what they do is assess different populations, microbiomes, or you can compare different treatment groups or something like that. So we know by using these beta diversity measurements that the microbiome of people who are obese or eat a Western diet are very reduced in diversity, as shown on the left. And we see an expansion of that Firmicutes phyla, which I spoke about before. So this is compared to the healthy microbiome, which is diverse and even, as I said before. Next. We also see an imbalance in the microbiome in diseases such as inflammatory bowel disease. So you can see the microbiome of this woman here with inflammatory bowel disease is reduced in diversity as well. And we see an expansion of proteobacteria in this case. And the species E. coli is a member of this particular bacterial phylum. And research in our lab suggests that this inflammation drives the reduction in diversity over time. And, but that's a talk for another day. Next. On the flip side, we know that hunter-gatherers have almost double the genetic diversity in their microbiome than the average American. So we see bacteria in their microbiomes that we don't see in people in industrialised countries such as Australia. So the blue star at the top represents treponema, which is found in the Hadza people in Tanzania, a hunter-gatherer population. Next. So it's important to note that there's temporal variation in the microbiome. Our microbiome is changing all the time. So if I asked you to eat a ton of high fibre food right now, then I can guarantee you that your microbiome now will look very different to what it will look like tomorrow. So we have to keep this in mind when we're doing studies. We can't just take one sample post-treatment, whatever we're looking at. We need to assess that temporal variation before the treatment. We also see seasonal variation in the gut microbiome, particularly in the hunter-gatherers who eat seasonal foods. They don't get it off the supermarket shelves. So the Hadza people eat a lot of honey and berries in the wet season and more meat and tubers and things in the dry season. And so their dry season seasonal microbiome is more like our microbiomes, but still distinct. So the Hadza people eat approximately 100 grams of fibre a day. And in Australia, we're encouraged as females to eat 25 grams a day and males to eat 30 grams a day. And only about 4 in 10 of us achieve that. And a high fibre diet is intimately linked with a highly diverse gut microbiome. So it's thought that their high fibre diet is responsible for this massive diversity, but also because they eat a huge variety of plant foods and whole foods. Next. So dietary fibre has many benefits. It increases satiety, so we're not as hungry as often. It's associated with decreased weight gain and therefore decreased propensity for cardiovascular disease, type 2 diabetes, etc. And it's associated with lower blood glucose and cholesterol. But the most important thing that fibre provides is that food for our bacteria. So the bacteria in our colon, our large bowel, ferment dietary fibre that we can't degrade ourselves, so insoluble fibre. And the byproduct of that fermentation is short-chain fatty acids. Next. So there are 3 main short-chain fatty acids that are produced in the gut butyrate, which is actually the main energy source of the cells lining your gut, so our colonocytes. 90% of the energy that those cells use is actually butyrate produced by bacteria. The second one is propionate, which has cholesterol lowering, anti-cancer and anti-inflammatory properties, but it's also associated with reduced fat storage. And then acetate, which regulates pH in the gut, potentially inhibiting pathogens, but it's also associated with increased satiety. So these short-chain fatty acids attach the cells of our gut walls, and then they move across the other side, and they end up in all sorts of areas, the target organs, including the brain, the liver, muscles, blood vessels, you name it. These short-chain fatty acids end up everywhere and maintain health. So there's no wonder that they're the source of much interest at the moment, and studies are looking at using them for the treatment of all sorts of diseases, including diabetes, inflammatory bowel disease, Parkinson's, asthma, and so many more. Next. So we can beneficially manipulate our microbiome, but there are a lot of myths out there as well. So I just wanted to break some of them down. We can manipulate our microbiome through diet. A high fibre diet, a diet that's very diverse, is going to lead to a very diverse microbiome, but not in the presence of a Western diet as well. There are better fats to eat, so those associated with a more Mediterranean diet, and they're the ones that we should be eating. Probiotics are also used to alter our gut microbiome, but they have limited success. So some people may benefit from probiotics, but others, they consume the probiotic and it just moves straight through them. So it's only having an effect, perhaps, while they're consuming those probiotics. Prebiotics are basically nutrients from our diet that we can feed bacteria in order to grow them up. And when you combine probiotics with prebiotics, so the bacteria with the nutrients that it eats, that's called symbiotics. And that provides the best chance for those strains to establish in the gut because they have the nutrients they need. There's some evidence to say that multiple strain preparations are better than single strain preparations, but again, more research is needed, particularly in the different areas. So there is other evidence as well showing that probiotics may actually delay restoration of the gut microbiome post-antibiotic therapy, and it may be more beneficial to put the microbes back that were there before than take probiotic supplements. So we're moving more towards a more personalised probiotic approach, I guess. So another technique for restoring the microbiome is one that I hope you've heard before, because I hope I'm not the first to introduce it to you, which is fecal microbiota transplant. Next. So this technique basically is used to reseed the gut microbiome, and it involves transferring somebody's own fecal material, so autologous FMT, or a donor's fecal material into the gastrointestinal tract. And it's not a new technique, it's been around since the 4th century, so Gi Hong, an alchemist in the 4th century in China, described a yellow soup, which was actually a fecal suspension. And Li Shizheng, a herbalist, Chinese herbalist, in the 16th century also described a fecal preparation that was used to treat diarrheal disease. So fecal microbiota transplant is being used in the clinic at the moment, but only to treat recurrent Clostridium difficile infection. So this infection can be life-threatening. People can go on to have pseudomembranous colitis, which may involve removing the colon or potentially death. But fecal microbiota transplant is effectively a cure for 90 to 95% of people with this recurrent infection. And this infection is commonly acquired in hospitals post-antibiotic treatment. Next. So there's a lot of ways that we can manipulate the microbiome. A lot of them are controversial, and there's a lot of basic studies in science being done to try and understand the interactions between the host and microbes. I just wanted to wrap up by introducing this study. It was actually done back in 2013, but it illustrates very well what I've been trying to get across today. So Redor et al., what they did was they took twins that were discordant for obesity, and they put their microbiome into germ-free mice, and the microbiome from the obese twin created an obese mouse. The microbiome from the lean twin resulted in a lean mouse. So then they took it one step further, next. And what they showed was that on a low-fat, high-fibre diet, so low-fat, high-fibre diet, the obese mice was ineffective at transferring its microbiome to the lean mice. So this is when the lean mouse and the obese mouse are co-housed. So they're coprophagic animals, so they'll eat each other's poo. So essentially a fecal microbiota transplant. So the fat mouse couldn't transfer its microbiome to the lean mouse, but the lean mouse could transfer its microbiome to the obese mouse, and it resulted in reduced fat content, reduced weight gain. And then the high fat, and then the same thing they did on a high fat diet, but the lean mouse was ineffective at transferring that microbiome to the obese mouse. So basically what this study suggests is that when we eat a high-fat diet, like a Western diet, this barricades any attempts to transfer microbiome or reseed the microbiome. Next. So I hope I've given you food for thought today. Thank you for listening, and I'll be happy to take questions after Wolfgang's talk. Thank you.
Thank you, Claire. We will take questions at the end. Just a nice place to stop, I think, now. I'd now like to introduce tonight's second speaker, Dr. Wolfgang Marx, who's joining us virtually tonight. Wolfgang is a joint Alfred Deakin and Multiple Sclerosis Research Australia postdoctoral researcher at the Food and Mood Centre at Deakin University. Wolfgang's research program covers a broad range of projects involving the use of nutrients, still, for mental health, fatigue, and cognition. Of particular interest is the role of polyphenols, compounds found abundantly in spices, fruits, and vegetables, in brain health and the gut microbiome. Wolf will speak for about 20 minutes. Thank you, Wolf. Please go ahead.
Hello, everyone. Thank you for the introduction. I suppose I'm the disembodied head of Dr. Wolfgang Marx. I'm a research fellow at Deakin's Food and Mood Centre, and tonight I wanted to provide an overview of the role that the food that we eat and the nutrients consume and how they relate to our mental and brain health. And I want to zoom in and talk a little bit more about the role of the gut microbiome in this process. And I put this slide up here because I think this is really, this is a great representation. And if you go to the next slide, I think it's just the perfect representation of our modern Australian diet, in that it's essentially we're eating too much of the stuff that's not so good for us, and we're consuming too little of the stuff that is good for us. So I think there might be a little animation there. Yeah, perfect. So we're essentially eating not enough of the stuff that's good for us, the fruits and vegetables, the whole food, unprocessed foods, and we're eating too much of the ultra processed foods, the processed meats, the high sodium, high fat, sugar sweetened beverages, and the like. If you go to the next slide, you'll see that it's no surprise that poor diet is the leading cause of premature death in the Western world, and this is second only to tobacco smoking and high blood pressure. if you go to the next slide, you can see that this has been slowly declining over the last couple of decades. So we can see this decline in our consumption of fresh green vegetables and a concomitant increase in highly processed convenience style meals. And this is really true across the socioeconomic spectrum. If you go to the next slide, you'll see that it's no surprise that we've come to this situation where more than 95% of Australians are not eating enough vegetables. So 4.4% of us are actually consuming the recommended servings of our vegetables. And if we see in the next slide, this situation is even more dire for our children. So just 0.4% of our children are meeting the minimum recommended number of serves of vegetables per day. And if you go to the next slide, it's no surprise that we are replacing all these really nutritious whole foods with ultra-processed foods. So this was a graph from a recent paper we published looking at the effects of ultra-processed food consumption on chronic diseases and depression. And when you look across various Western data sets, we can see that at the top we have 58% of Americans' energy intake is derived from ultra-processed foods, and this is fairly consistent across Western cultures. And while Australia isn't up there, we know that we are no exception to this rule. So going to the next slide. So I guess that's a snapshot of our diet, and sorry to start on a downer, but I suppose the summary there is that our current state of our diet quality is not so great. Now, if we look at the other side of the coin of this presentation, I want to give a similar snapshot of the current state of mental health across the globe. So in our next slide, we can see that this is the chart of the global burden of disease attributed to various chronic disease categories. And you can see in that kind of light orange color, this is neuropsychiatric diseases. So you can see that the vast sort of, or the largest proportion of global burden is attributed to neuropsychiatric disorders, more so than things such as cardiovascular disease and cancers. And if we zoom in on this on the next slide, you can see that it's really our common mental disorders, so that is depression and anxiety, that's really playing the largest role in this global burden of disease. On our next slide, so there's obviously more work that needs to be done, more effective treatments we need to look into to help manage this increasing global burden of disease attributed to mental illness. And one of the exciting areas that I think that is developing over the last decade or so is this renewed interest in the role of modifiable environmental and lifestyle factors, and designing interventions around these factors to help improve our mental health. And one of those is, of course, diet being a modifiable risk factor. So in this next section, and if we change slide, I want to talk about what we know so far about the role of nutrition in our mental health. So in our next slide, we can see that if we look at observational studies, if we go to the next slide, sorry. Next slide. Yes, sorry. So if we look at what we know from observational studies, so where we look at really large data sets of populations and we examine the association between one factor and another, we know that a healthy diet, be it a Mediterranean diet, adherence to a healthy dietary guidelines, a traditional Japanese diet, a Norwegian diet, however you quantify it, is associated with a reduced risk of depressive symptoms. And what's promising is that we see this not only in adulthood, but across the lifespan. However, what we know is that this is a promising support for a hypothesis, but if you've heard of the saying, correlation does not always equal causation, what we need to do is we need to take this information further and look at it through randomized controlled trials. And this is exactly what we did. We ran, if you go to the next slide, you'll see the details of the SMILES trial. This was a 12-week intervention in people with clinical depression, and we randomized them to either receive a nutrition intervention or a non-dietary social support control group. And if you go to the next slide, you can see at the end of those 12 weeks, we saw this really significant drop in reported depressive symptoms, so much so that at 12 weeks, 32% of those in the dietary support group achieved remission criteria. And what's exciting about these results is that they have also been replicated by other researchers in their own randomized controlled trials, both within Australia and abroad. If you go to the next slide. So now that we have this data that suggests that diet as an intervention can be effective in treating our mental health, the next question then goes to, well, how might diet impact our mental health? And this is a figure from a recent publication of ours where we proposed a number of different mechanisms that seem to be affected by diet and that may improve our mental health. And of those, we identified the gut microbiome as being a particularly promising area of research and a particularly promising pathway that may link this diet and disease paradigm. So if you go to the next slide. So I guess just to reiterate what Claire said, and I think, I guess if we had the same event maybe 10, 15 years ago, Claire and I probably wouldn't have that much to talk about. It's really been in the last 10 to 15 years this explosion of research that's been investigating the role of the bacteria within our gastrointestinal tract and how that relates to chronic disease outcomes. And if you go to the next slide, you can see that this communication between our gut and our brain has spawned a whole host of different interesting research questions. And so this communication between our gut and our brain is termed the gut-brain axis. And as you can see on this figure, it's not a one-way street. We consider it a bidirectional communication pathway where the bacteria within our gut communicates with our brain, but also our brain seems to communicate with our gut. And this bidirectional process has been investigated for a whole host of different outcomes, including epilepsy, stroke, pain, neurological disorders, and of course, psychiatric disorders such as depression. So if we go to the next slide, I want to talk a little bit about why we think the gut microbiota might be involved in our mental health, specifically in depression. So if we measure the microbiome of people with depression and compare that to otherwise healthy controls, as a number of studies have, we can see that there seems to be differences in this microbiome. There are some bacteria that are more prevalent in people with depression, and there's some that are less prevalent in depression. And this points towards the direction of, well, maybe there's something going on here. But of course, it's a bidirectional process, right? So maybe the depression is causing a change in microbiome. Maybe the medication people are taking are altering the microbiome. Maybe the change in lifestyle and behaviors are altering it. So it's really kind of the first piece of the puzzle, but we need to look at it in different angles to see whether it's more of a causal pathway. And as you can see in the next slide, this is one of the really interesting tools that we have. And this is a particular mouse model that is a germ-free mouse. So this is a mouse that was essentially born without a microbiota. And this mouse model has essentially been integral to the microbiome field in general in teasing out the causality and the role of the microbiome in various different processes. But in particular to brain health and mental health, we can see if we look at the germ-free mice and we compare it to mice with an intact gut microbiome, we see all these really interesting and stark differences. We can see that the brain volume is different. We can see that they have different levels of neurotransmitters and things such as like serotonin. Their myelination is different, which is relevant to people with multiple sclerosis. All these really interesting things that point towards, well, maybe it's the gut microbiome that's driving part of this disease process. But I think one of the more compelling and, to me, kind of one of the coolest experiments that have been done in this area, if you look at the next slide, similar to what Claire mentioned with obesity, we have found that if you recruit people with depression and then a group without depression, and we transfer that gut microbiota into these germ-free mice, those mice that receive the gut microbiota from those with depression then start to display depression behaviors. So we're essentially kind of transferring depression, which kind of seems a little bit science fiction, but it's a really compelling case, I think, to the causality and the involvement of the gut microbiome in driving this process. And now if we go to the next slide. So now that we have this feeling that the gut microbiota can potentially drive some of this process, of course the next question is, well, how can we modify the gut microbiota to then use it as a treatment tool to improve our mental health? And as Claire mentioned, there are a whole host of things that can influence our gut microbiome. Principal among that is the role of diet. And I guess just to add on to and just second what Claire said, if you go to the next slide, this is just to say that it's not about. Well, I guess what we see with the role of diet on the gut microbiota is that there's this rapid shift in our gut microbiota composition in response to a change in diet. It's not a situation where we're consuming a particular diet for months on end and then there might be gradual shifts. This is a study that showed that there were shifts in as small as 5 days, but of course there are studies that have shown that there's been a rapid shift in as small as 24 hours. And I think that's a really important message, that we're really seeing this constantly shifting gut microbiome and how powerful the diet can be as a potential treatment tool. If you go to the next slide. So what I want to talk about is the role of what we've termed now psychobiotics, which is essentially dietary-related interventions that target the gut microbiota with an aim to improve our mental and brain health. And what I want to do is start in the next slide on the role of probiotics. So as Claire mentioned, probiotics are essentially beneficial bacteria that we can consume. So we know when we go into Chemist Warehouse and such, we see the whole row of the different probiotic supplements. But of course, it's important to remember that these probiotic bacteria are also present in the foods that we consume. So things like yogurt and kefir, sauerkraut, kombucha, these fermented products as well. And if we go to the next slide, we can see that there's been a recent review that has looked at the randomized controlled trial literature that have investigated the use of probiotic interventions for depressive and anxiety symptoms. And when they pull all of these different studies together, we can see that the conclusion is that there does seem to be a significant decrease in depressive symptoms and anxiety symptoms when consuming these probiotic interventions. What's also promising is that we see that there's a greater effect in people with clinical depression compared to those without a clinical diagnosis, which I think is a great thing because, of course, the people with the clinical diagnosis are the people that are in most need of relief. But similar to what Claire said, I'm also in the camp that, maybe it's a little bit early days. There are huge levels of inter-individual differences. All our microbiotas are quite different and our response to probiotics are quite different. We also need long-term safety data to ensure that these are actually safe to consume. Now I want to briefly talk about prebiotics in the next slide. Oh, sorry, before I get to that, I also just wanted to briefly mention that there's also been some research in the use of probiotics for other mental health disorders as well. So this was a really interesting study that looked at rates of rehospitalization in people with acute mania. And as you can see from this graph, you see this really nice separation in the rate of rehospitalization in people that received a probiotic compared to those in the placebo group. Now we'll talk about prebiotics. So Claire talked about the role of dietary fiber as a prebiotic, and I won't go through that again, but I think one of the really interesting parts of this area is that we're also learning new ways in which the different dietary nutrients may interact with the gut microbiome and provide beneficial properties. So if you go to the next slide, what I mean by that is omega-3 fatty acids, which are considered it by some international guidelines to be a second-line therapy for depression. You know, we have a really strong evidence base for the use of omega-3 fatty acids for depression. But what's quite interesting is that we're now finding that these may actually have prebiotic properties. So they're not only acting through the pathways that we have known and have studied for quite a long time, but they also may be acting through our gut microbiome. Similar to this, if you go to the next slide, is the role of polyphenols. So these are essentially pigments or compounds within a diverse range of whole foods. So when we think of why extra virgin olive oil is good for us, or dark chocolate, or red wine, or blueberries, we're essentially talking about the polyphenol content of these foods. And what we're finding now is that high consumption of polyphenols appear to be related to a lower risk of depressive symptoms. And if you go to our next slide, you'll see that this beneficial effect seems to be really strongly driven by the gut microbiome. And so this relationship between polyphenols and the gut microbiome is quite intricate. So the polyphenols, they seem to have prebiotic properties which increase our abundance of beneficial bacteria, but our gut bacteria seem to metabolize these polyphenols and may be actually responsible for some of their beneficial properties. If you go to the next slide. So I'll just quickly show a nice example, I think, of this. And it's difficult without the mouse, but essentially this was a mouse study that in one group, the mice received a high-fat diet and they measured their blood glucose. And they see this spike, which is signified by the black line. In a second group, they received a high-fat diet, but they also received polyphenols. And they saw this blunted response in blood glucose, which is a good thing. But in a third group, they received the high-fat diet, the polyphenols, and an antibiotic, which essentially wiped out their gut microbiota. And when they did that, you can see that they actually had this really marked spike in their blood glucose, which demonstrates that there's this really strong relationship between the gut microbiota and polyphenols. And if you go to the next slide, I'll briefly just mention that the sort of last term, the last sort of psychobiotic area is the role of fermented foods. And I think this is really an understudied area because the fermented foods not only have the probiotics that we talked about, the prebiotics that we talked about, but they also have these third class of compounds called biogenics, which might have their own immune stimulating anti-inflammatory properties, which essentially have all three in one food item, which I think might be a really powerful intervention. Unfortunately, at the moment, there just isn't any good clinical research to support the use in mental health, but I think this is an area of upcoming research. So, in the final two slides, I think, because I threw a lot at you in these 20 minutes, And so I think it's always good to sort of come back around to what we can do to incorporate some of these lessons into our diet. And broadly speaking, I think it revolves around the inclusion of fermented foods in our diet, consuming a diverse range of plant foods to get that fiber, but also those polyphenols and the various types of fibers to improve our gut microbiome. And on the other side of the coin, reducing our intake of those ultra-processed foods. Next slide. And that's, I think we can skip that. I'll just briefly finish up. If you're interested in learning more about the role of food and mood in the gut microbiome, we have a free online course which you can find on Google and on our website. And if you're interested in following us, I have, in our last slide, I have the website as well as our Twitter details if you'd like to learn more. Thank you very much.
Thank you so much, Wolf. I would now like to invite Claire and TJ back to the stage for question time with our audiences. For those who are watching online, please submit your questions by scanning the QR code or click the link on the page where you're watching the stream. If you are in the Shine Dome, please do make your way to the microphone. However, you might have heard there is a pandemic on, so please do keep social distancing in mind as you do this. While everyone makes their way to the microphone, I'm going to go ahead and ask the first question from our online audience. Wolf and Claire, I'm going to ask this to both of you. We've had lots of people in the online questions asking about specific conditions and what they can do to either treat or prevent or lessen the symptoms from those very specific conditions with treating their gut health. Now, it's not fair, obviously, to ask you to give people individual dietary advice online, but what would you say to those people? I'll start with you, Wolf. If someone's looking for help with depression, with one of the other mental health conditions that you mentioned, what would you recommend they do in terms of addressing their gut health?
Yeah, sure. So I think this is a really important question. And I think while we're learning so much about how the gut microbiome can influence our mental health, and it's getting more and more complex. I think when we zoom out on the sort of take-home messages, it really falls back to just improving that diet quality. So as I said in that final slide, it's really about including those fermented foods, those diverse range of whole foods, things like beans and spices, which as Australians we don't quite get enough of compared to other cultures. These sorts of things. It's not necessarily around specific diets or restrictions or anything like that. But I will say, if you are having a hard time managing the dietary aspect, the best port of call is really to speak with a dietitian. There are now dietitians that really specialize in that sort of food-mood connection, and they can really help you individualize your diet and make sure that it's something it's sustainable and evidence-based.
Thanks, Wolf. Claire, would you like to add to that?
Yeah, sure. Obviously, I'm not a dietitian, so the first thing I'd say is talk to a dietitian if you're looking at changing your diet and you have, something like inflammatory bowel disease, which is the disease state that I study most. But in terms of the microbiome, yeah, it's exactly what Wolf said. It's a large variety of whole foods, high fibre, that's going to really increase the diversity of your microbiome. And you really need to work with a dietitian if you have inflammatory bowel disease to work out what that diet might look like for you, because there's so many complications. These, people with inflammatory bowel diseases have strictures and ulceration and all sorts of things. So, I think the first point of call would be a dietitian who will be able to work with you to try and incorporate as many foods as possible.
So I'm hearing no silver bullet and see a dietitian.
Well said, well said.
TJ, I believe you have a question there in the Shine Dome.
Yes, we have a question here, and I'd invite the audience to come up and ask questions. The microphone here. Go ahead. Speak, try again.
Yeah, so the question was from the audience, do people who are larger in size but don't eat a Western diet also have an altered microbiome? Look, the answer is yes. Usually an obese person will have a microbiome that's reduced in diversity. We can actually transfer our microbiome to our children. So like I said, babies born vaginally will acquire mostly their mother's microbiome. So if the mother's obese, if she's eating a Western diet, she's had recent antibiotic use, so we know that antibiotic use associated with C-sections is also detrimental to the microbiome that's transferred to the baby, i.e. it's not there. But also hormones and things like that can dictate our microbiome. So polycystic ovary syndrome is an example of where the microbes, the hormones and the endocrine system can really affect the microbiome in a bidirectional manner.
Your second question?
Thanks. A little bit more practical. You both mentioned diversity of foods and the microbiota, and I was wondering if, on a practical level, you'd recommend trying different brands of yogurt because they all seem to have different strains of bacteria in them.
Yes, so it depends on the yogurt. Some yogurts don't contain enough of the good bacteria to be beneficial, and it's a very personal thing. So if there's a niche in your microbiome that microbe can fall into and become a resident, then that's great. But you know, I think we don't know when we eat it whether it's going to establish in our gut or not. So I guess, yeah, the preparations that include multiple strains are probably more likely to establish in our gut. But, you know, we all know that eating a diverse diet and a high fibre diet is associated with higher diversity, so that's probably a better approach. But I do eat a lot of yogurt.
We'll go to an online question now. Wolf, I'll ask this to you first. Do you think that gut microbes are linked to sleep and mental health? Obviously, we know sleep and mental health are connected. Could our sleep be affecting our gut microbes, or could our gut microbes be affecting our sleep?
The answer to that is yes, I think. I guess what we're seeing in many chronic diseases and disorders and the such, we're seeing that it's really this bidirectional thing. We're seeing that, particularly with mood and stress, that seems to influence our gut microbiome, and the gut microbiome seems to influence our stress response. I'm sure, although I'm not a sleep expert or knowledgeable in the area of insomnia, I'm sure that there's this bidirectional process that's showing up there as well.
Awesome, thank you. TJ, I believe you have a question in the dome.
Yes, we have another question here.
Thanks. Thank you very much. My question is, before 5-6 years back, we used to, all the media or all the research used to say, if you've got any disease, more or less 70 to 80% is because of your genetics. But now, altogether it's changed. Now, what I can see on the really some scientific papers where they are mentioning it's more like 20 to 30%, but 70 to 80% is through your microbiome, through your guts. So what's your view on that? Thanks.
I think that was directed at me. Sorry, Wolfgang, I can't see your face on the screen to see whether you were going to take that one or not. Yeah, a lot of the diseases we've solved that aren't, that involve just an infectious organism or are just genetic. So now we're left with all these complex ones. So Crohn's disease, which I study, for example, there's definitely a genetic component. We know there's susceptibility loci that encode for genes that are involved in bacterial sensing and things like that, and killing of bacteria. So it's this, we're left with all the complex diseases to solve, and I think that's why the microbiome's been implicated more and more in them these days. Yeah.
And the microbiome has its own genetics as well, of course. And they're part of us, as I think Claire mentioned earlier too.
Yeah, we've definitely evolved with them, and sometimes we're not doing them any favours with all the antibiotic use and things like that. We're driving them to their limits as well, and so they're, fighting to survive just like we are. And so we're a product of that combined evolution with them, and I think that's why we're hearing this bidirectional all the time, where we exist in a symbiosis with them. So. Yeah, we need to treat them with respect.
So one question for Wolfgang and one for Claire. Wolfgang, we had a speaker from Headspace last night talking about the number of youth having depressive and suicidal thoughts. Is this something that we could alleviate with changing their diet?
Yeah, so I think that's a really important point. Most of the age of onset for most common mental disorders is in our young adolescence. And so if we can, discover more effective treatments and adjunctive treatments that we can use, we might be averting a huge proportion of that global burden of disease that I was talking about. Most of the interventional research has been in adults, but there has been a recently published study that looked at dietary interventions in, young adults, so university-age students, and they found similar results to what we found in our adult clinical trial. It's certainly an area that requires further research, but I don't see any reason why the results that we're seeing in adults wouldn't be transferable to those in young adults. And really, at the end of the day, it's about improving diet quality. So whether it improves, I guess there's no harm in improving our diet quality, because at the very least we're improving our physical health as well, which is obviously something very important too.
And Claire, is there any room for people with irritable bowel syndrome in your study?
Yes. As I said, I study the inflammatory bowel diseases. They're often confused with irritable bowel syndrome. So irritable bowel syndrome is still a significant issue for a lot of people. It doesn't involve inflammation of the gut like inflammatory bowel disease, but it can cause some awful symptoms and things like that. But definitely, there has actually been some studies showing that fecal microbiota transplant may be beneficial in that group of people, and probiotics as well. But again, it's not a one-size-fits-all, and I think we need to move towards more personalised probiotics and medications. A lot of these sorts of therapies are under, they're being made at the moment. There's, companies popping up around the world, and what they're doing is they're culturing the microbes from our microbiome. So previously we were only able to culture about 70% of the microbes from our gut microbiome. So people are working out the specific recipes for growing these bacteria now, the nutrients that they require, the oxygen concentrations and salt concentrations that will allow them to grow in the lab, and they're putting them in capsules. And so I think the probiotic space will vastly improve in the future.
I have a question for both Claire and Wolfgang. Taking us back to the first talk about genetically modified foods and sort of the benefits and some of the ethical considerations, do you see any potential benefits with using genetically modified foods, or do you have any concerns? Is there any potential in this space for pre or probiotics and genetic modification of foods?
That's for you.
This might be a good one for TJ, actually. He works more in the genetically modified space. Again, sorry, Wolfgang, I can't see your response, your facial expressions. But genetically modified foods, it's a highly regulated field. And obviously there's like the CSIRO, I know have put out some good cereal products and things with a high fibre content that work. They're often used in studies, they're often fed to mice, and so these products can do what they purport to do. I guess people do get scared when you mention genetically modified, but if it's, like I said, it's highly regulated. I don't have an issue with them, but I'll let Wolfgang respond in turn.
Yeah, no, I'm of the same vein. I think that's a great example. I guess the products that increase the fiber content. I think that, for some people, the availability of fresh foods and those sorts of things is not always an option. It can be difficult, can be expensive. Potential strategies around that that still offer that nutrition, I think, is a good thing. But if that targets areas along the gut microbiome, I think in general that's a good thing.
I'll just link, obviously we're linking the last Food for Thought series when the omega-3 fatty acids were mentioned, and Wolf mentioned those tonight as well. So that was a very nice link as well. Thank you for that.
It is a series. I have one last question for you. This will be the last one in the interest of time. This one's come through online. Obviously, COVID-19 has put infectious diseases at the front of everyone's mind. What role do you see the microbiome having for infectious diseases, either in the gut or outside of the gut? Wolf, do you want to go first?
Sure. So, the gut microbiome plays an integral role in our immune response, particularly for the food that we consume and such. So that can't be denied. I would not posit a guess in terms of how that might relate to, COVID and the rest. I think there's enough noise online for me to muddy any waters. So I think I'll just leave it there.
Claire, would you like to try with that topical question?
Yeah, like Wolfgang said, the microbiome trains our immune system, and one study I think I'd like to see done is the effect of the hygienic environment we're forced to live in at the moment on the establishment of the gut microbiome. So the whole idea of the baby's born by C-section and not acquiring that normal vaginal microbiome and things like that ties in with the hygiene hypothesis, and it'd be interesting to see if there's an increase, a little spike in things like asthma and allergies in the future in babies being born right now. So we're all socially distancing and using our hand rub and trying not to transmit anything. Yeah, I'm sure somebody's doing the study.
I hope so. Well, thank you so much, Claire and Wolf, for joining us tonight. We really appreciate your time and your energy. Thank you to the audience as well for joining us tonight. And I also need to thank our event sponsors, Gira Wines at Gira Station and Hoberg Catering. And also thank you to the Shine Dome team that have made it possible for us to host this hybrid event across Australia tonight. Thank you all for joining us. We do hope to see you again in August for our next talk in this series, which is on alternative food sources. So thank you and good night.
Very good.
it's something I prescribe. Yeah, it's something I prescribe to, and